Okay, so I'm going to go ahead and get started. I think my screen should be showing, hopefully. Hopefully everyone can see my screen. It says that it's sharing it. If it's not, please feel free to type in and let me know. Like, I think there's a way to ask a question or reply to questions. Let me know if for some reason you don't see my screen, but it should be showing my slides. So I'm going to go ahead and jump right in. So first of all, just welcome to everybody. Thanks for tuning in tonight, and I will make this recording available afterwards once I get it all processed. I will have some time at the end for questions, so if you do have questions about anything we cover, you can certainly type those in, and we can go over that once this is all over. So the first installment of Gut Chat, hopefully this is something that I can do, monthly to kind of cover topics that people have some interest in, where I'm really hoping I'm going to be able to make this super complex topic at least a little bit less intimidating for those of you listening, because I know this is very heavy on the science and information, and it is a bit complicated. So some of the information I'll present tonight is a little bit complicated and high level, like some of the research studies I'll reference maybe, but I'm going to do my best to break that down, what exactly the utility of that information is, what does it mean for the rest of us, right? So hopefully it's a little bit easier to digest, pun intended. All right, so let's jump into what you can expect tonight. The topic is diversity, and this topic, like all others relating to the gut microbiome, is quite heavy, and we're really only starting to scratch the surface of how this operates in humans. So we're in this great period of time in research where it's just now starting to explode on the scientific landscape in human studies. So I'll start out by defining exactly what this concept of diversity means, talk about how it has shaped us lifelong. And a lot of factors that shape diversity are things we can't control at all, but others are things we certainly do have control over. So next I'll talk about what keystone microbes are, what that means, how they affect diversity, and then we'll talk about what this does, or what does it all actually mean for our human health. So things like disease, obesity, medical conditions, things that have been linked to microbiome diversity. And then last, we'll talk about what can we even do about diversity if it's low. So what is microbiome diversity? Well, the good thing is it's something we can actually measure and test for. So it's not just this theoretical concept, but it has many layers to it. Most of the time when we think about diversity in the microbiome, or the term diversity in general, whether it's people or animal species, we're thinking about how many different types of something exist in a set. So in this case, how many different species do we see in someone's microbiome? This part of diversity is referring to the richness of the microbiome, which is, this is important that more species we have present, the more adaptive and flexible our microbiome is. So that if there's maybe a major episode of illness or an inflammatory exposure, like a foodborne pathogen, right, we're better able to return to what we refer to as homeostasis, or a state of normalcy or health, as close as we can get to that, versus in cases where there's less diversity in a person, for instance, if they have very low diversity in their microbiome, a really inflammatory episode, whatever that may be, might set that person back for much, much longer before they recover any kind of normal balance, if at all. And then we also consider the evenness of the diversity. So this concept is looking for there to be balance among all the species represented with an established kind of quote-unquote normal parameters. So not that we need to see equal amounts of each bacteria because their populations are not meant to be equal, but that we don't want extreme dominance of the microbiome from certain species that shouldn't be found in high abundance. So for instance, bifidobacterium only makes up about 5% of the microbiome of adults, but it's considered a keystone bacteria with a great amount of influence. If its population were to fall to perhaps 1 to 2% of the microbiome, then other bacteria that we don't want much of could then grow more abundant in its place because it's missing, and those become more dominant in the ecosystem than they should be. So it's not even. The key takeaway here, this is sort of a 60,000-foot view of someone's microbiome and diversity. So this is just one part of like the testing, for instance, that I run on patients. It gives us some clues, but it's not going to break it down into super deep detail yet. However, from this, I can glean a lot of information about how their microbes are distributed, what types of imbalances we might find in species-specific panels later on in the testing. And then this gives me a lot of information that I can ask questions and dive into potential history and causes, or maybe it gives me clues for what to look for next. So the key takeaway here is that there are layers to balance. We want to see all the different angles here and balance, not just one or two, and not all variables are affected by the same things. One thing I want to point out over on the far right, the Firmicutes to Bacteroidetes ratio, so where it says F to B, that, it's a lot to say. You can just call it the F to B ratio for short. And this is going to come up. This ratio is really, really interconnected to a lot of health outcomes. A lot of the research that's being done out there in both animal and human populations has focused heavily on how this ratio changes and responds to different things. You're going to hear it quite a few times over this webinar, so I just want to make sure I indicate how important it is right now. This is referring to the ratio of two of the largest phyla in the gut microbiome. So the two largest groups of bacterial distribution within each of these, within Firmicutes and within Bacteroidetes, there are many, many, many different species, thousands of them. These two groups have sort of a healthy range that they each need to be in, but then they also need to be in a certain ratio to each other because the species within each of them need to be in balance with each other. And when they're not, we see everything from obesity to insulin resistance to autoimmune disease, intestinal dysfunction, inflammatory diseases, neurological disorders, on and on and on. So this is a really foundational concept to the microbiome when we talk about diversity, and it's been researched pretty heavily at this point. All right, so then the interventions being different for, how is our diversity impacted? Is the richness low? Is the evenness low? Interventions would be different for each person, but then we also have the causes. So with the causes being different for each of these, depending on the patterns we see on lab tests and symptoms and things like that, which we're going to dive into next as far as the causes underlying this, this kind of gives us a way to take a look at how this person might be presenting and what types of things might we see in somebody who has their diversity impacted. Okay, so what impacts diversity? All right, the next few slides are going to be kind of information heavy, but I've tried to summarize the important findings. Just keep in mind that there's tons and tons and tons of studies out there done in animal populations that have looked at far more than what we've been able to study in humans in just the last decade or so. Basically, everything that affects the diversity of the microbiome from even before we're born, everything affects it, it seems. Now this slide and the next couple fall into the category of things you can't actually do much about, but you still need to be aware of, because what has happened to you in the past still impacts your current and your future. And then for those of you who plan to have children or have more children, it's pretty important to keep these things in mind. So first, dads, sorry, but you don't have a ton of influence on a child's microbiome in utero, but after birth you will, so just be patient. Moms, though, kind of everything you're doing is impacting your unborn child's microbiome. Instead of freaking out and thinking you've got to make everything perfect, let me just caution you that the dirtier your surroundings, the more diverse that baby's microbiome is actually going to be. So studies are actually showing that moms who are exposed to farm animals and other animals during pregnancy have children who have significantly lower prevalence of allergies, autism, asthma, and autoimmune diseases, the 4 big A's, compared to moms who live in a very sterile environment. So moms that are constantly cleaning, using lots of household cleaners, they're kind of germaphobes, they're kind of freaked out about germs during pregnancy, their children tend to have more of those inflammatory disorders later on in life after birth And a lot of it goes back to acquiring this diverse microbiome in utero. It also, mom's diet is heavily influencing the baby's microbiome. If mom eats a Western diet that's relatively high fat, and when we say Western diet, sometimes that means high fat, high sugar, but sometimes it just means high fat. So even there's some smaller studies that have even shown like ketogenic diets during pregnancy still have the same effect as far as changing the microbiome of the baby. A higher fat content diet, regardless, it affects the microbiome by changing the F to B ratio pretty significantly in most cases. And those children are more likely to be overweight or obese in childhood. And then you'll hear me mention antibiotic use a lot tonight, but antibiotic use by mothers during pregnancy has lasting effects on the microbiomes of their children, even after birth, which has been shown to lead to higher incidence of allergies, eczema, and hay fever, which are kind of that allergic response category. Now, really quick, what's really profound about that concept is that when you are in utero, before you're born, so that baby that's mom's pregnant, baby's in there, that infant or that unborn baby's gut has no microbes in it. It's considered basically sterile. So you're not colonized by any microbes until after birth. So even though you have no microbes to kill off with those antibiotics before you're born that your mom takes, the antibiotics your mom takes during pregnancy still can kill off or alter the survivability of microbes you are exposed to and acquire after birth, which is actually pretty astounding. I didn't know that. That was actually something new I learned as far as the fact that these antibiotics have lasting effects after birth. All right. Okay, so now that you're born, yay! But the birth method is now really important. So if you're born vaginally, you're going to be colonized by a completely different set of microbes than if you're born via cesarean birth. The vaginal canal is full of lactobacilli bacteria that are all, for the most part, pretty helpful and beneficial. So as the baby's pushed out through the birth canal, it's acquiring all those good lactobacilli on its skin, mouth, lungs. Those microbes make their way into the gut and start to flourish, especially when the baby starts to breastfeed, which is rich in naturally occurring sugars that feed those good bacteria. So the lactose that's present in human breast milk is a food source for those early bacteria. But if you're born via C-section, you're not only not exposed to all the vaginal microbiome, but you're going to be colonized by the microbes in the operating room and on the skin, the outer skin, obviously, or obviously skin is outer, the skin of your mom, which are predominantly Clostridia, Streptococcus, and Staphylococcus. You'll have lower lactobacillus levels, which will tend to lead to greater risk for asthma and some allergies in childhood. And then there's also some connection with greater risk for overweight or obesity in those kids too. Again, altering that F to B ratio early in life. Now, preterm or premature infants are a whole different story because even if they're born vaginally, which some are, but some aren't, they're kept in these really sterile environments, which has some benefits, but may also contribute to some reduction in diversity too. So it's kind of, weighing the pros and cons. The biggest risk for these babies, though, is that most of them are given a lot of antibiotics, and I mean a lot. And some of those are life-saving. So I'm not saying let's stop antibiotics for preterm babies, but let's absolutely realize that what we're doing for that child later in life with complications and medical risks that may occur. So again, weigh the pros and cons. Medicine is fantastic, and it has made tremendous advances. And I think that, the fact that we're able to deliver babies that are in some cases 3 and 4 months premature and keep them alive, and they're able to live, mostly normal lives as far as they eventually reach their growth milestones and all the things they're supposed to do, that is remarkable. So please don't take that as a comment on we shouldn't be giving them antibiotics, because some of them need it for sure. All right, so then we have infants. Let's say that you survive pregnancy and childbirth and your microbiome's okay. It can still be affected by a lot of things. The primary influence at this stage is diet, and that's dependent heavily on the source of nutrition, so whether you're breastfed or formula fed. Human breast milk is rich in bifidobacterium species, which are transmitted to the baby, and which make up to 95% of the gut microbiome of breastfed infants. These bifidobacteria, which you're going to hear a lot about throughout this webinar, are absolutely important species, and you'll see them pop up over and over tonight. Basically, we really, really want to acquire these as soon as possible after birth. If we don't, then we're going to be colonized by more Clostridia species early in life, which means an altered F to B ratio, and higher risk for overweight or obesity, as well as possibly other inflammatory conditions. All right, so then we have the sibling effect. This is a really neat finding from microbiome research that has shown that if you have at least one older sibling, your microbiome is more diverse, and as early as 5 weeks of age, we see this enhanced diversity. Even if you're not vaginally delivered or breastfed, that diversity won't be equal to someone who was vaginally delivered and breastfed, but it will still be higher than someone who is an only child and has some of these other diversity risks. So the reason for this is kids are walking germ factories. So your older sibling is constantly transferring microbes to you, whether they're oral, fecal, or otherwise. And yeah, it's pretty gross to think about that, but in the grand scheme of things, your older brother or sister's poop germs actually made your microbiome better. So just be grateful, try not to think about it too much. Another thing that's being pretty heavily studied is environmental exposures of all kinds. And you'll hear this also come up as kind of a theme tonight, including pollution. And what we're finding is that babies and children who are born into and grow up in areas with greater air pollution to urban areas typically have really altered gut microbiome diversity, especially that F to B ratio, and then more incidence of inflammatory diseases later in life, including obesity, autism, and autoimmune disorders. So we're already starting to kind of connect some of these dots here in research. Now, I'd also like to just plant this seed right now that this is probably one of the major contributors to why we see so much more prevalence of a variety of childhood disorders, not so much because we're better at detecting and diagnosing them, but because now in today's world, our world and environment have become more and more polluted. So the pollution now is significantly worse than it was 10 years ago, let alone 20, 30, 40 years ago. A lot of which we have not a lot of control over, right? Our microbial defense mechanisms are not evolving as fast as the pollution in our environment is occurring. Air pollution is just one way this happens. There's also groundwater, tap water, food, crops, contaminants, industrial chemicals, that are used in clothing, manufacturing, housewares, so on. So I won't go off into the weeds on that, but just consider that our microbiome hasn't yet adapted to these new threats as fast as these threats have developed in our lives. All right, so not going to go in depth on every single one of these, but take a look at just the sampling of factors that start to hit our microbiome as we enter childhood. Antibiotics, who hasn't had a lot of those in childhood, right? Especially if your parents were the type to take you to the doctor's office for every sniffle, even though most of the time the antibiotics you were given were unnecessary. And then what if they were really necessary, like ear infections or tonsillitis or recurring bronchitis? So some kids are actually getting a lot of antibiotics in childhood, and they are usually much sicker as adults, and they have much more imbalanced gut health as an adult. Whether or not you're around other germ-ridden children in preschool or daycare is pretty big. Please make sure you vaccinate your kids before sending them off to school, letting them play around other kids, but send them off to school or even play groups to help build their microbiome diversity. Pets are huge, right? Having a pet in the home makes our diversity better because pets are also pretty germ-ridden. So they help us acquire some of those bugs. I already talked a little bit about whether we live in urban areas or rural areas. We're getting exposed to completely different microbes. A lot of that's air quality. So we're going to have more diversity living in a rural or even semi-rural environment in childhood. Lots of dirt, outdoors, bugs and stuff. If we don't eat enough fiber as a kid, we can impact diversity, which most kids are picky. And they're not eating a ton of fiber, but at the end of this webinar, I'll list some foods that can impact some of these major groups we're going to talk about. And some of them are definitely kid-friendly, so there's always something you can do. Secondhand smoke, it shouldn't have to be said, but yeah, don't let your kids be around secondhand smoke ever. If you smoke around them, stop. If other relatives do, get them out of that situation for lots of reasons. And I briefly touched a little bit on environmental exposure to things like pollution, but we also see the flip side of positive exposures like being on a farm or around farm animals and fresh air, which is actually full of bacteria, by the way, which colonize first in our airways, but then later on in our gut. And we have much lower incidence of asthma if we're breathing that outdoor, but clean outdoor air, right? So try to get away from the city often with your kids if you do live in the urban areas. Once we make it to adolescence and our teenage years, this is another big milestone where we start to see some imbalances occur. And maybe it's because teenagers eat like crap, or maybe because of puberty, or maybe because of their environment, but we do know some things that are likely to develop. First, if people smoke or use tobacco products, that is often when they are first exposed firsthand, is during teenage years or adolescence. Using tobacco exposes us to quite a few pathogens that are known to reside on the actual tobacco leaves. This was something I didn't know, something new I learned putting this together. So we're just ingesting these really inflammatory bacteria there, but then also cigarette smoke itself alters the signaling of immune cells, which is part of why cigarettes are so inflammatory. It sort of has this domino effect down to reducing normal immune responses and allows our immune system to sort of prune away certain beneficial bacteria, which allows pro-inflammatory bacteria to thrive in their place. There's not a lot of human studies on recreational drugs in the gut microbiome, believe it or not, and that's a field that's just now in its infancy. Most studies are looking at the effects now of THC or CBD, both cannabinoids, and the gut microbiome, and most of those studies show at least some benefit to supporting diversity, or they're maybe neutral. So there's not really a benefit, but there's not a harm in using them as far as diversity goes. There are some considerations about the cleanliness and processing practices of actual marijuana plants that might contribute to some contamination, though, when using that. So that's just something to kind of throw out there. Puberty absolutely does start to exert an effect on the microbiome, and it's through various hormone signaling pathways that this primarily occurs. So testosterone and estrogen both signal differently in the microbiome, and that's why we see so much difference in inflammatory diseases with links to the gut in women, which we'll get to here shortly. The gut-brain connection, though, begins to be really, it's there, obviously, before puberty. We really start to see it a little bit more prevalently or obviously here in the gut. And we also now know that this is where we see, again, not that they're more prevalent mood and psychiatric disorders in teenagers or adolescents, just as they begin to become more obvious. Perhaps, maybe just because we start to pay a little bit more attention to them as people. Before that, we just consider them kids. And then, of course, diet. Teenagers don't eat the best. So part of that also has to do with the fact that they tend to eat lower fiber diet, and some of the alterations to their microbiome, if they never really develop better eating habits like eating plants, fruits, vegetables, etc., those can actually become permanent or a little bit longer lasting if they don't begin to reintroduce better foods at some point. Okay, and then last, here we are at adulting, and pretty much the same things affect the adult microbiome that affect infants and children, only by now your body's much closer to death and you feel the effects of it more, right? So we're still using a lot of antibiotics, and those are still wiping out our bifidobacterium and lactobacillus. Both, remember, super important groups, altering that F to B ratio, and we're even more prone to weight gain and autoimmune conditions because our body isn't as adept and efficient at actively renewing itself, removing all the toxic byproducts of our adult life decisions. We're still being exposed to a lot of environmental junk that we can't always control. This really interesting study here, right there on that second bullet, found that geography and environment exposures actually independently affected diversity in both vegans and omnivores, where we would have expected to see the diversity be pretty different between vegans and omnivores. Vegans obviously eating only plants, no animal products, and omnivores eating both plants and animals. But it turned out that it was only different based on environmental exposures. So this kind of tells us, again, our environment probably has much more impact on us than we think it does at this point. And then last, as you get even closer to death, your family is probably going to put you in an old folks home because they're not into wiping your butt. And this is virtually stealing your death warrant because studies of all kinds, not just on the microbiome, show that older adults who live in their communities, so like they live with their family or they live on their own, they're not in an old folks home, live much, much longer than those in homes or long-term care. And one facet of that is microbiome diversity and immunity that comes from it. They're less likely to get sick, and when they do get sick, it's less likely to hospitalize them compared to those in care facilities. Again, environment's huge here, pollution in our water, home, and then, hey, sexual activity, right? If you think toddlers are germ-ridden, most adults are too, possibly more so, not always with germs that are going to help our diversity, right? So we actually acquire the microbiome patterns of people we have sexual intercourse with. Sometimes that means sexually transmitted infections. Sometimes it just means normal microbes, so it's not always necessarily a bad thing. To a certain extent, we acquire microbiomes that are similar. Obviously, genitals are going to vary. Women are going to have different microbiomes vaginally, obviously, because the environment is different. This can be both bad and good, right? So animal studies have actually shown that the genital microbiome of males can influence genetic expression in their offspring. So how healthy they are and what microbes live in their genitals, basically in the glands that actually make their sperm. So it's not talking about like the outside of their skin, but inside, can actually change the genetic expression in their sperm, which is very interesting. Again, our food is quite polluted with a lot of stuff. A lot of that stuff is super damaging to the microbiome and its diversity, including but not limited to the obvious pesticides, which that's what pesticides are designed to kill. Microbes are part of that. Also, though, plasticizers, other various additives in both food, but also food packaging, which a lot of us aren't always thinking about. But you remember not too long ago when BPA, right, bisphenol A, was finally ruled to be really dangerous, and how long it took the FDA and EPA to recognize that. And so then finally, hospitalizations. Even if you're not that close to death, just being hospitalized for something routine or mundane, like, a simple procedure, or perhaps you just have an episode where you are hospitalized for a few days, puts you at pretty big risk. They've shown that within 48 hours of admission, people pick up all kinds of hospital microbes, which tend to be more inflammatory and even pathogenic, like Clostridium difficile. Most of you have probably heard of C. diff, and that's what that is, Clostridium difficile. In immunocompromised people, especially, who have been given a lot of antibiotics in the hospital, it can kill them because their healthy microbiome has been wiped out so thoroughly by those antibiotics. This C. diff comes right in and causes acute diarrhea, which eventually leads to death through organ failure from nutrient depletion and dehydration. So not a fun time. All right, so now we know what can cause low diversity, but let's talk a little bit about what microbes we should see in a diverse microbiome. So these are kind of the main groups. Big words, you don't have to recognize them or remember them, but I kind of want to throw this out there because some of these, as research gets better and better and better, I think you will start to see more and more of these, even in like random normal conversation. Like, I think most people have heard the words bifidobacterium and lactobacillus if they've ever bought a probiotic before, because those are the two most probably common ones. But we're now starting to see some of these other bacteria show up in probiotic products, at least the aerobic ones. If they're anaerobic, you're probably never going to see them in a supplement form because they can't survive an oxygenated environment. But then also note down there at the bottom under proteobacteria, right? This is a category that is really inflammatory. These are the ones where you're going to see your like really bad foodborne pathogens, E. coli, Yersinia, which is the plague bacteria, Shigella, enteropathogenic E. coli, so on and so forth. So the good news is we can test for all of these. All of these are things that we can find and detect in microbiome testing, and we do, in fact, and these are the things that we want to look for as far as the balance of these and the ratios to each other. All right, so what are keystone species? This is really, really central to the concept of diversity, so I kind of want to explain what this means when I say keystone species. A really good way to think about keystone species is that they perform a very specific function, and without them present or abundant enough in the microbiome, the host, which is us in this case, would experience some kind of negative consequence or side effect. So this could range from just some inflammation when we eat, or manifest as ulcerative colitis, maybe nutrient deficiencies in the case of bacillus species to help us make vitamin K2. Basically, they have a disproportionate amount of influence on the health of the host, meaning these are usually not bacteria that are, a large percentage of our microbiome. So before I mentioned bifidobacteria, right? The totality of all bifidobacteria make up about 5% of the microbiome of an adult in the U.S., which isn't that much when you think about it. Out of, you know, an entire 100%, we've only got 5% that are bifidobacteria, but of that, 5% from that bifidobacteria is huge. If that were to drop to 4%, that would make a very big impact on the health of the host and perhaps, you know, create a very symptomatic type of inflammation for that person. So again, disproportionate amount of influence. We're looking for these bacteria or microbes that fill a very specific niche, and without them, imbalance and inflammation happens pretty radically. Okay, so what keystone species should we have? Well, there's not really a good answer to this question because human diet varies widely across geography, culture, ethnicity, age, and so many more variables. This graph depicts the varying effects of dietary influences on a human-derived microbiome, and this was a pretty interesting study. So it's not technically a human study because the researchers transplanted human gut microbiota into germ-free mice, which means the mice had no microbes in their gut at all. They were completely sterile. Then they had human fecal transplant, which basically installed a gut microbiome from a human into the mice. The reason why they used mice and not just humans was because they wanted to see this, how it evolved over several generations. As we know, generations of mice are significantly shorter than humans. So it's a much shorter study, basically, as they reproduce much more quickly, I guess, how that microbiome changed over time. And then also it's very difficult to do this in humans because humans are prone to really, really erroneous reporting errors when it comes to dietary intake. So when I ask people, and this has actually been studied when researchers or clinicians ask patients what they've eaten, patients are either deliberately or accidentally misleading or erroneous, and it can be up to 60% incorrect, which is huge. It completely invalidates an entire study when it is patient self-reported data in many cases, because humans are so inaccurate, apparently, when it comes to dietary intake reporting. So there's no way to completely control all the environmental variables that humans are exposed to also. So again, we've already talked about all of the exposures of the environment, right, that affect our diversity, whereas rats in a controlled environment, we can control that, obviously. So it would take a very long time to collect the data. It would be hard to control all the variables. So one thing I do want to point out here on the picture, just to kind of understand what you're looking at, is the LFPP stands for low-fat plant polysaccharide rich, which means the diet was probably Mediterranean-like or pescatarian-like in terms of macronutrients in humans. That would be what its closest pattern would be in human populations. The Western diet was defined as high sugar, high fat. So you can see that some really distinct patterns emerge. The switch to a Western diet produces a drop in Bacteroidetes and an increase in Firmicutes. So again, that S to B ratio, that key ratio that predicts risk for obesity and metabolic syndrome. Then there's also a really interesting finding from the study that points out that regardless of the host diet, so the fecal transplant from whoever, or I'm sorry, the host, the animal host, whether it was the low-fat plant polysaccharide rich or Western, within 7 days of implanting into the donor microbiome, the distribution and diversity took on that common and predictable pattern based on the recipient's diet, not the original microbiome from the donor. So what that means is that whoever the donor was, whatever the human was who donated the fecal sample, right, whatever their diet was, the host's diet or the donor diet, the rat that it was transplanted into, their diet then changed the diversity of the microbiome transplant. So even if it was from somebody eating a Western diet, a high-fat, high-sugar diet, transplanted into the rat, if the rat was put on a low-fat plant polysaccharide-rich diet, that diet was actually going to reflect in that microbiome within 7 days of the transplant. So that's actually pretty interesting. We know the microbiome diversity is very dynamic and it can change rapidly. So that falls in line with previous research. And then on that point of obesity, the humanized microbiome mice switched to a Western diet from the low-fat plant polysaccharide rich. So if they started on the sort of Mediterranean plant-rich diet and then they were switched to a Western diet, they did in fact retain more body fat, or their adiposity increased, we would say, within 30 days of that diet change. So it did actually induce a body fat accumulation in those mice. And then another really interesting finding from that same study, which has been found in other studies too, was that some of the species that bloomed under the Western diet conditions actually showed greater genetic transcription of genes associated with simple carbohydrate digestion. So translated into layperson, that means these microbes are more efficient at extracting calories from simple carbohydrate foods, which is one of the theories as to why they promote obesity. However, I think there are some other causes that go along with this as well, such as gram-negative bacteria, which we're going to talk about coming up. But just think about that. When the microbiome changes and that diversity is impaired, and we see that diet influence the microbiome, and it selects for bacteria that upregulate genes that now extract more calories from carbohydrates being eaten, we're actually absorbing more calories from our diet, which is part of why potentially we are gaining more weight. So just kind of a little bit mind-blowing. Now, here's a study that's been widely cited for its look into the microbiome of one of the last true hunter-gatherer societies of humans. This is the Hadza tribe in Tanzania, Africa. They're kind of a microbiologist's dream because it provides a glimpse into what ancestral human microbiomes look like. Now, an important thing to remember when talking about ancestral microbiomes is that they most likely did not all look like this because their diets were very different depending on available flora and fauna. So European ancestral microbiomes probably look very different, and the Inuits and ancestral humans inhabiting the Arctic and Siberia looked very different still. The Hadza diets consist of 5 main categories of food, being meat, honey, baobab, berries, and tubers. So they don't farm or breed any type of animal. They are completely non-agrarian. When you look at them compared to urban living Italians, which is the control group, they're considered the Western diet here as well by all macro and micronutrient standards. Kind of a Mediterranean diet pattern as far as the micronutrients. You can see the really striking difference in richness of species or diversity in the Hadzas compared to the Italians on a Western diet. So that's the first little color graph there. You can see all the little different colors represent a distinct taxonomic or distinct species or taxa of bacteria. So you can see how in the Hadza there are so many more colors, and then in the Italians, which are considered the control group or the Western diet, you see the same 3 or 4 maybe that dominate, and that's about it. So there's much less diversity here in comparison. Now, the macronutrient composition of the Hadzas is said to be about equal to that of the Western diet in terms of just comparing straight up carbs, protein, and fat. So this is where I think some people have argued in the past, well, maybe it's because they're eating more, fiber, less carbs, different proteins, so something like that. However, they did show that the macronutrient composition was relatively similar. They eat significantly more fibrous root vegetables, though, so their fiber intake is much higher, even though carbohydrate intake is comparable. More of the carbohydrates in the Western diet are low fiber, so pastas and rice and things like that. Now, one of the reasons I hesitate to say that a hunter-gatherer diet is the basis for a normal diversity pattern is that the Hadza had basically non-existent bifidobacterium, Bacteroides, Glaucia, Lactospiraceae, Rosaburia, Fecalibacterium. All of these are considered very beneficial and critical anti-inflammatory microbes in humans. Again, you don't have to remember that, but these were all microbes that were essentially missing from the Hadza that we see as anti-inflammatory in Western populations. So maybe the Hadza don't need these because they don't eat foods that require these microbes' assistance. Maybe these microbes only show up in populations that have certain dietary patterns that necessitate their presence. They also had a much higher prevalence of microbes that are considered very opportunistic or actually inflammatory in Western individuals, which means in this particular society, perhaps under the right dietary conditions, these microbes are at least neutral, if not symbiotic. So I think that we have to be very careful drawing conclusions or drawing correlations between a hunter-gatherer society and a Western society because, again, environment is very, very different. And then also what's cool is that that same study pulled out comparisons of prior research into other hunter-gatherer societies that have been studied, as well as other westernized societies. And you can see there are definitely some patterns that you'd find in a sort of ancestral versus modern diet. Now, whether or not this is purely because of what we eat, I would argue is not really going to be supported. And in fact, there's that study from previously in the webinar from Wu, 2016, that actually demonstrated that the exposome, or environmental exposures, have significant impact on what our microbiome looks like. So think about hunter-gatherer tribes. They're not exposed to the air pollution, the water pollution, the textile manufacturing, fuels, munitions, paint, VOCs, etc. Their environment is relatively clean from synthetic and manufactured pollutants, despite having lots and lots and lots of different microbes, some of them pathogenic. They have very different patterns, and I think that has a lot to do with it. There's likely a lot of the differences above in the charts attributed to the fact that the Western patterns are influenced by that pollution, pesticide, indoor living, chemicals in our, clothes, furniture, cleaning supplies, vehicle emissions, you name it, right? So we're being hit by a lot of different stuff, and our microbiomes are responding accordingly to that, whereas these hunter-gatherer populations are not experiencing that. Now, what's cool, and I didn't include any research here, but you can actually look at the research that shows that when you take someone from an environment like a hunter-gatherer society and you transplant them, like let's say that they immigrate to a Western country or an urban area, within one generation, their microbiome begins to mirror that of the people who live in that area. Again, a lot of it is environmental exposure, but they begin to develop allergies, asthma, autoimmune, and autism at the same rates that those urbanized populations do as well. So we can't say that it is purely genetic. We can't say that it's purely diet. We can't say that it's purely anything, any one thing, but we do know that environment has a lot to do with it. It's definitely not all diet. Okay, and then here's an illustration from a random 5 patients. This is, again, another way to look at diversity. These are 5 patients I've worked with, I've run in-depth testing on. I didn't put up any of the individual panels, but I wanted to kind of just show the little diversity chart. Diversity falls in those main categories. So now it's important to note on this slide that this is a bit of a selection bias because all of these people sought me out to work with me on various GI symptoms or disorders, so I can't say that any of them had a normal microbiome or a balanced microbiome. But you can see that even here in these charts, there's a pattern that emerges. The F to B ratio, the blue and purple sections you see on there, is that ratio that predicts obesity, as well as generally higher inflammatory conditions and a less stable immune response. And that ratio should be somewhere around 1-to-1 or 2-to-1. And then the proteobacteria, which in the West are the most inflammatory and tend to be pathogens, should be pretty small. And look at some of the patterns where the purple or Bacteroidetes are very low and the proteobacteria have kind of taken over that niche in their place. So it's kind of neat to see how these groups are all interrelated here. When one group goes missing, another is going to take its place, and it's usually not another helpful bacteria that does that. And then finally, it's often postulated that vegetarians and vegans have better, quote-unquote, microbiomes or more diverse microbiomes because they eat a greater proportion of calories from plants. Therefore, they've got higher fiber intake. And while they may have different distributions of bacteria, it turns out that their diversity is actually not any discernibly different from omnivores, people who eat both plants and animals. So now where it should be noted that you will see a difference is in the blood levels of metabolites that come from plant polyphenols, which is a type of plant phytonutrient, which means the microbiome of vegans yields greater levels of these plant-based antioxidants simply because there's greater consumption of the plants from which they come. So the consumption of animal proteins is not associated with those polyphenols, which we find that even if you eat animal protein, you would still get the antioxidant benefits of the vegan diet if you ate a similar amount of plants. So if you remember my comment earlier on about eat like a vegetarian that eats animals, that's kind of the goal here. All right, so what keystone species should we have? We've looked at all this evidence, all these charts, all these, theories. And the answer is that nobody really knows. And there's probably isn't a quote-unquote diverse microbiome that's appropriate for all of us. Because remember, we have this really intricate relationship between our environment, our genes, which we haven't even really talked about and we won't get into tonight, our microbiome. So what's diverse for me won't be diverse for someone else with different circumstances. The best that we can do is attempt to restore what feels like balance to each individual. So we're going to test them, see what they're missing, try to restore that, and bring things back into balance. And this can take time. So it's not something that happens overnight typically. Now to the meat and potatoes. A lot of this information on how diversity impacts health we've touched on briefly in the past sections throughout the life cycle, so I'm not going to be too verbose here, but I just want to present some kind of striking findings. All right, so you can see from this really simplified diagram the causes of loss of diversity on the left and the effects of loss of diversity on the right. I just want to point out one thing before anyone asks. Right now, there are no really good randomized controlled clinical trials that definitively say something on the left absolutely causes something on the right, and it's because of the loss of diversity. So we can't quite make that connection, but we have RCTs, randomized controlled clinical trials, that show that the stuff on the left causes low diversity. And then we have correlational or epidemiological data that show that low diversity is found in all of those things on the right, or can lead to those things on the right, because of both the length of the trial needed as well as the variables that would be impossible to control in human populations, which I already kind of touched on. Humans are pretty inaccurate at reporting things. Not to mention the ethical clearance needed. There's probably no way we'll ever really be able to design a trial where we can actually say that this is the case. So that's just kind of an FYI. That's why a lot of the research is done in animals, because we can actually set up the exact conditions we need to be able to show that. And that is a limitation, unfortunately. But again, autoimmune disease, neurodegeneration, metabolic abnormalities, cancer, neuropsychiatric disorders, those are all things that have been connected to low microbiome diversity or loss of diversity, and it's for a number of reasons. One of the themes you're probably picking up tonight is this connection with diversity and overweight or obesity. So here's how this happens. Now first, not all overweight or obese persons have low diversity, although many do. According to data trends, however, not all overweight or obese cases are due to diversity alterations, and they're not all completely due to that. Obesity is a very multifactorial thing, and like diversity, it has many layers. But the things we do know are that when we wipe out or significantly reduce lactobacilli and bifidobacterium populations through whatever means we've done that through, we tend to see more gram-negative bacteria. Gram-negative refers to the type of cell wall and the specific little markers on the bacterial cell walls that are kind of like signaling molecules. These bacteria have differently structured cell walls, and that's part of why there are different types of antibiotics, because some antibiotics work on some bacterial cell walls while others need something completely different to penetrate the type of cell wall and kill the bacteria. So gram-negative bacteria aren't all bad per se, but we need them to be balanced by gram-positive bacteria. Gram-negative bacteria, though, produce endotoxins, which are little chemicals that they make and are highly inflammatory to humans. These endotoxins, and depending on the virulence or how aggressive that bacteria is, their endotoxins can cause, vomiting, diarrhea, nausea, cramping. Those are the type of endotoxins produced by foodborne pathogens. They cause really, really violent reactions in the human host. And then there's gram-negative bacteria who produce endotoxins that are not outright foodborne pathogens, but they're still pretty inflammatory, and they do definitely provoke and upset our immune system and cause inflammation. These endotoxins, referred to as lipopolysaccharide, or LPS, you can refer to it as LPS for short, they disrupt the membrane proteins in our intestinal lining. So the little proteins that sit and hold our intestinal cells together, those membrane proteins are actually disrupted when LPS comes in contact with them. And then LPS can kind of sneak into our systemic circulation or leak through that barrier. This is referred to as metabolic endotoxemia, And we've started to see a lot of research connecting this piece to the advancement or promotion of inflammation, then atherosclerosis, then non-alcoholic fatty liver disease, then metabolic syndrome. And each one of those little progressions shows exactly what LPS is doing. So it's not just that it triggers inflammation, it's that it's also doing these other things too. So it's been pretty widely studied in animal populations as a cause and effect, and now we're starting to see some human trials that are beginning to show the same mechanism, albeit in pieces still. What we have known for some time is that people with diabetes and metabolic syndrome have different microbiome patterns and definitely less diversity. Again, the F to B ratio is altered. They've got less bacteria that makes these really beneficial compounds called short-chain fatty acids, which butyrate is mentioned there in the second bullet. These compounds, like butyrate, actually regulate our intestinal immune response. They keep our intestinal barrier nice and tight, closed up, and without the bacteria that make them, like lactobacillus or bifidobacteria, some of the other species that are mentioned, like rosaburia there, fecalibacteria, also make butyrate. We're kind of left to fend for ourselves. So there's these bacteria that make this really beneficial compound that helps to fight this entire sequence. When we kill off these bacteria, again, through whatever means they're being killed off and suppressed, we don't have this control mechanism, and so it kind of goes haywire, and there's nothing stopping this progression of inflammation at that point. In this scenario, part of what we also see with the metabolic endotoxemia is that it actually changes the membrane uptake of sugar and amino acids, and this is what's really important here, which changes insulin response. So there appears to be a protective mechanism of butyrate. Again, these bacteria that are really great and beneficial make butyrate. These bacteria make it, and it acts protective against developing type 2 diabetes in part. So really, really neat research starting to come out and kind of pick apart this relationship. Another part of this is, on the next slide, there's a picture of this. I know this is wordy and it gets a little bit dry. This is a lot of words to say that there's conversation going on between bad bacteria in the gut, which are overgrown or too abundant, because our good bacteria aren't thriving. We have killed them off with whatever we've done. And these bad bacteria are telling our signaling receptors, our toll-like receptors, they're little signaling receptors that pick up on everything. They kind of sense everything going on. They're all along the gut as well as all over the body, but we're talking about the ones in the gut. They will actually, these bad bacteria are actually communicating and having a conversation with these receptors, telling those receptors to make us feel hungry, right? They stimulate our appetite. So we eat more and we take in more calories. They're also interfering with the signals going to our pancreas, which alters insulin response. So we have more incidence of insulin resistance occurring. And then to top it all off, that endotoxin, LPS, that I mentioned on the previous slide, is still there. And when it's present, it's actually, in addition to sort of sneaking through that intestinal barrier, the LPS that's inside the gastrointestinal tract is pinging off of these toll-like receptors, and it's continuing this cycle. So it's constantly stimulating those receptors, stimulating appetite. We eat more calories. It's interfering with insulin signaling or insulin production in the pancreas, etc. So it kind of creates a self-perpetuating cycle when we have this loss of diversity and the wrong bacteria dominating. Sorry, I had to just drink some water. So the last slides I mentioned that not only do we have these bad bacteria there sending pro-inflammatory signals, but they're there because we have lost so many of our good keystone or beneficial bacteria that would normally keep the bad guys under control. There's actually quite a few negative side effects to the loss of those keystone bacteria, which leads to overgrowth or dominance in gram-negative bacteria primarily, producing that LPS. This LPS actually disrupts the proteins in the tight junctions. We talked about that. They break apart from the tight junction and leave that junction wide open. So think about like breaking the gate to your backyard. They break the gate down, your backyard is wide open, so anybody can come in, right? LPS gets into the bloodstream. If this continues to happen repeatedly or chronically, this is that metabolic endotoxemia. So you can kind of see in the diagram, it's circulating now in the body, in the bloodstream. It's not acute. It's not like acute endotoxemia, because there is a condition that people can be hospitalized for when they have a really aggressive pathogen present somewhere in the body, not just in the gut, but it could be an infected wound. It's what's part of sepsis. That's endotoxemia. But metabolic endotoxemia is sort of this low-grade inflammation that's just enough to provoke the immune system. So what's not shown here in detail, this is a very, very, very simplified diagram, is there's a number of immune responses and immune cells like CD14 cells, T cells, et cetera, that are constantly being triggered and turned on and fighting as this LPS continues to circulate. And what often happens is, in addition to this, is those cells start to become dysfunctional and they misread immune signals, and they can begin to attack body tissues, so like autoimmunity, essentially. They do, in fact, cause damage to insulin signaling in the pancreas, so there's actually damage to some of the beta cells in the pancreas that occurs because of this LPS. So that's one form, but this can also happen with rheumatoid arthritis, Hashimoto's thyroiditis, etc. This also causes oxidation of cholesterol, which leads to more plaques in the arteries. And so that's also something to kind of keep in mind, that with this metabolic endotoxemia, we've actually seen that there's more oxidized LDL, which is deposited into the blood vessel walls, which is atherosclerosis, and it's directly because of LPS, because our cholesterol particles, most people don't realize cholesterol particles are fairly good. Their job in the body, in addition to transporting fats around, is they're part of a detoxification mechanism. So the liver will actually send LDL particles and sometimes HDL out to grab up things that are fat soluble, and LPS is one of those. So we often actually see cholesterol levels go up in individuals with this metabolic endotoxemia. And the reason why is because the liver is trying to control the levels of this particularly toxic compound that's getting into the body. And when you deal with the overgrowth or infection in the gut, you actually usually see cholesterol return to normal. And in fact, I've seen that on labs time and time and time again. So if you do happen to have seen lately, like if you've had a cholesterol panel run with your doctor and it's higher than it's normally been, and you've got some like a little bit of inflammation in the gut, consider that the cholesterol is probably a downstream effect of the inflammation going on in your gut. You probably have some reduced diversity, probably some overgrowth of gram-negative bacteria. Deal with it. Don't just let it go on, because it can become something much worse in the future. All right, so I don't want to get too far off track. Here's a really neat study that showed that by simply changing the diversity of the microbiome of obese subjects, this was actually in humans, which was neat, obese subjects with metabolic syndrome, these individuals' inflammatory markers were improved over a 23-week period. So you can see in the upper left, I won't go into each one of these categories of bacteria, but in the upper left, the changes in proteobacteria, which, remember, that phyla is many, many pathogenic bacteria. Those are like E. coli, Shigella, think foodborne pathogens, and then there's sketchy, slightly less inflammatory, but still bad for you relatives. That category, they produce a lot of that LPS endotoxin, okay? And then in these individuals, they found that those people had sort of that low-level metabolic endotoxemia. The actinobacteria, the one right next to it, that phyla is what bifidobacteria belongs to. That phyla actually improved and went up, which is really neat to see. I won't dive into all of the other ones listed. Overall, they show a decrease in problematic or inflammatory bacteria. So if we see like the gamma proteobacteria, some of the other ones, they increase keystone beneficial bacteria, which corresponded with improved inflammatory markers, improved glucose, and glucose sensitivity, and improved insulin control. Some human trials that are just now starting to look at this stuff that we've been studying in animal models for a really long time, implanting lean microbiota into obese individuals improves their insulin sensitivity. So this is a fecal transplant. It selects for better diversity in those individuals. Multiple sclerosis and rheumatoid arthritis, both autoimmune diseases, have very different diversity compared to healthy controls. So we're learning ways that we can affect diversity potentially to change the outcome of those diseases. And then there's even some microbes that have been shown to affect the genetic expression of genes that alter autoimmune risk. So those genes in the HLA family, if you're familiar with genetics and you've heard of HLA-DQ, those are the genes related to celiac disease. This is the same family as celiac and type 1 diabetes genes. The HLA family of genes basically affects other autoimmune conditions as well. And then this was just kind of a cool resource I found. I won't dive into this too much, gender differences in the microbiome diversity. And it all has to do with what hormones are dominant. So it has very much to do with the hormonal milieu and effects going on and how those are communicating with the bacteria in our gut. One of the big things to note is that females have a very different F-to-B ratio compared to males, which is part of what is speculated to contribute to the increased incidence of autoimmunity in women. That and testosterone is thought to confer some protective or modulating effects on immune signaling in men, which is a very different conversation, not necessarily related to microbiome diversity. Last but not least, so the question you're all asking at this point is, what can we even do about low diversity? Because it sounds like it's hopeless and we're all going to die of it, right? Well, it turns out that we can influence this, and this is a bit pedestrian. It's going to be much more specific and detailed based on testing for what I would actually have one individual person do. But these are kind of the big things that all of us will respond to. You'll notice I don't have specific probiotics listed here because that really needs to be individualized based on someone's lab testing. But what we do know that despite all things we're going to make it hard for bifidobacteria to thrive, and that's just life in general at this point, we can feed them anthocyanin-rich fruits, which those are fruits with a blue or purple color to them. Anthocyanin is a polyphenol associated with blues and purples in plants. This polyphenol is like bifidobacteria's favorite meal, and it's been shown to improve abundance of that bifidobacteria. There's also the red polyphenols in wine and purple, black, and red fruits. Please don't overuse antibiotics. They're meant for life-saving situations, not the common cold. They usually don't even work on the common cold. Try to avoid heavily pesticide-laden foods, and bifidobacteria, appears to be disproportionately affected by those pesticides. That's why. And they're used in common farming in the U.S. at least. Eat a lot of plants, and I mean a lot of plants. Again, I've said that my husband and I have always said to clients, eat like a vegetarian who eats meat. So really prioritize a variety of plants of all colors. All plants matter, not the poisonous ones, obviously. Breastfeed your babies if you haven't had them already. If you're planning to, and for whatever reason you cannot breastfeed, because that does happen, and that's not always in your control, there are breast milk donation services. You can obtain fresh breast milk from other moms who donate, whose babies no longer need it. I highly recommend you look into that if you need it. It is that important. Lactobacillus responds well also to red and purple polyphenol compounds. So again, red wine, grapes, apple skin, pomegranate, cherries, raspberries, blackberries, currants. Pectin-rich foods like apples, pears, and citrus, FOS fiber, there's some examples there. Again, don't overuse antibiotics. These guys can be easily wiped out with those as well. Same thing with pesticides. Many of our lactobacilli species are heavily impacted by these especially. And then if at all possibly, don't just decide to have a C-section if it's not medically necessary. Believe it or not, this happens. Obviously, you can't control circumstances that require a C-section birth, and please don't feel guilty or ashamed if you have to have one. But there are ways to help your baby acquire the right microbes if you do have to do that. So again, it's not the end of the world, but some women do actually choose to schedule C-sections because it's more convenient for them to plan the birth of their child around other life events. It's just, if it's not medically necessary, it's not really recommended. And then finally, improve your exposome. Remember, I used that word before. The exposome is all the exposures we have around us at all times, some good, some bad. Antibacterial hand gels are pretty bad for your microbiome. Yes, even your internal microbiome. If you followed the news in the last few years, you may have seen where the triclosan that used to be in those antibacterial hand gels is actually banned. It's supposed to be phased out. And partly because it is so damaging to the microbiome, it leads to all kinds of immune imbalance in people who use those products. Harsh cleaners in your home or workplace can often contain compounds that negatively affect diversity. They absorb through the skin, those types of things. Personal care products, same thing. Eat more plants, but spend time around more plants and spend time around animals. Their germs are good for you. Spend time outdoors where you can access fresh air, dirt, plants, animals, sunshine. All of that has been linked to improved microbiome diversity, believe it or not. And then just finally, limit exposure to VOCs, which stands for volatile organic compounds. These come from things like fuel, paints, waste facilities, smelting, welding, manufacturing, so on and so forth. They often get into our air, leach into our groundwater. So some of those things we can't necessarily control, but if you happen to have a craft or a hobby that involves some of those things, just be aware that that might just be an additional risk that you're taking on in doing that if you happen to be somebody who has some sensitive diversity in their microbiome. And I think that is it.